Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Hello, dear ladies and gentlemen. In today's lecture, we will discuss about chemistry for physical amino acid and protein. In the human organism, protein undertake multiple and vital functions. Structural protein are found in each and every cellular compartment. Intracellular motor protein power the energy-dependent transportation of vehicles. Signal protein receive and transmit information, transport and storage protein bind, for example, oxygen and carry it to a location where it is needed. Of equally great significance are enzyme protein, as without these biocatalysts, a smooth and frictionless in exchange of energy and nutrient would be practically impossible. This article illustrates the role of amino acid as a building blocks in the structure of protein, their chemical properties, and also present the ways in which amino acid fulfill their multiple duties. General structure of amino acid: the residue makes the difference. One can safely deduce the component of an amino acid from its name. They include carboxylic acid port, namely a port that contains a carboxylic functional group. Then comes an amino acid on the left side of the alpha carbon atom, which is the second carbon atom to the carbon atom which is with the highest oxidation state. In this case, it is the carbon atom of the carboxylic acid in a strict sense. This is an alpha L amino acid. Next to the carboxylic amine group, a hydrogen atom in an R side chain. R is used to convey the meaning of residue of R root. R subsequence attached to the alpha carbon atom. Each amino acid contains a different side chain which define their specific properties as we shown in the figure. This is a simple structure of the amino acid. Protogenetic amino acids are those that can be found in human beings. Currently, the scientific community is familiar with 21 different amino acids which can be divided as follows: non-polar chain, glycine, alanine, valine, leucine, isoleucine, methionine, proline, phenylalanine, and tryptophan. Non-charged polar side chain are trypsin, serine, threonine, cytosine, selenocytosine, asparagine, and glutamine. Acidic side chain are aspartate and glutamate. Basic side chain are lysine, arginine, and histidine. The essential or indispensable amino acids are those that have to be supplied to the human organism through the diet, since they cannot be synthesized by it. Those include the essential or indispensable amino acids are those that have to supply to human organism through the diet, since they cannot be synthesized by it. Those include methionine, threonine, valine, histidine, tryptophan, phenylalanine, isoleucine, leucine, and lysine. Essential amino acid that cannot be synthesized in the body. These amino acids are required in the diet as we shown in the figure. Different amino acids are present in this figure. Overview: protogenic amino acids are chemical properties of amino acid are: amino acids are amphoteric molecules. This means that they possess both basic as well as acidic properties and therefore constitute very good buffer system. Their functional group are the reason for this. The amino acid has a pre-unbound pair of electrons on the N atom, and so it can function in a basic way. It can accept a proton. On the other hand, the carboxylic group release a proton due to its positive electrical charge of the H atom, thus functioning in an acidic way. In this way, an individual molecule can at the same time assume both the rule of proton donor and the proton acceptor. The intracellular proteolytic reaction lead to the formation of Euterion, namely compound containing carboxylate and ammonium group, which are firmly bound together in a crystal lattice. They are distinguishable by their high melting temperature and good water solubility. Analogically to ionic bond, Euterion take their name from the German Zweiter, meaning hermaphrodite or hybrid, as we shown different type of Zweiter in the following figure. The first one is alpha amino acid, dipolar structure Euterion, predominant at pH1, predominant at pH 7, predominant at pH 11. In water solution, the pH value is of vital importance. When the pH value is that of isoelectric point, i.e. Phe specific pH value, amino acids are present in the form of Euterion. If the pH value of the solution is below the IEF acidic solution, amino acid gain a proton and become cation. The proton is gained by the N atom of the amino acid, which are as a result become an ammonium group respectively. They form anion when the solution pH value is greater than that of IEF. In this case, the greatly available hydroxide ion take a proton away from the carboxylic group. This leads to a deprotonated carboxylate group. The graphic depiction of this pH-dependent behavior result in the titration curve of an amino acid. In very low pH value, amino acid exists in the protonated form, ammonium form, conjugated acid cation. As the pH value rises, even more carboxylic group dispose of their proton. When the pH reach to pKs1 value, the concentration of the cationic form equal that of Euterion ionic form. If the pH value rises above the pKs1 value, this Euterion predominate in the concentration. When pH is equal to IEF, only Euterion are found. When the further rise of pH value, the ammonium group also disposes of its proton. The concentration of an ionic form build up until the pKs2 value is reached. Then again the concentration of Euterion match that of anion. In cases of very high pH value, only carboxylate conjugated bases anions are to be found in a solution. This is the highest deprotonated form of amino acid. The pKs and IEF value are characteristic in each case. If even more protonable or deprotonate side chain are present, pKs3 and pKs4 value are additionally be described. Typical reaction of amino acid are the decarboxylation of an amino acid lead to the formation of biogenic primary amines and CO2. These undertake vital transport function, e.g. catecholamines like adrenaline and noradrenaline are built from trypsin and phenylalanine. The vital inhibitory transmitter GABA result from glutamate. A further example is a histidine, from which histamine is built. Histamine is a hormone produced in the body tissue and is responsible, amongst other, for immediate or hypersensitivity reaction. The transmission is a reaction through which an amino acid 1 reacts with a keto acid 2 catalyzed by the enzyme aminotransferase. As we can understand from its name, the amine group is exchanged. The amino acid 1 original amino acid convert to alpha keto acid, and the alpha keto acid to convert to amino acid 2. Non-oxidative deamination are also eliminating. Deamination involves the removal of water from a certain amino acid. The formation of unstable intermediate product lead to alpha keto acid and ammonia. The three aforementioned reaction always require PLP as a cofactor. On the other hand, the fourth typical reaction that amino acid engage in the oxidative deamination require water and NAD instead. The latter is downscaled to NADPH divided by H positive. Just like the elimination, deamination, the final product here is ammonia, and the corresponding alpha keto acid, amino acid to protein are, as we shown in the figure, the peptide bond between the amino acids. This is a human insulin structure. A protein can be formed in thousands of amino acid residues. The properties of the substituent are responsible not only for the intramolecular but also for the intramolecular interaction. When two amino acids are bound together in so-called peptide bond, dipeptide are formed, and should another one be added, a tripeptide. Oligopeptide consists of up to 10 amino acids, whereas polypeptide of over 10 amino acids. At the stage when polypeptide conjugate, a protein is produced with 100 to 1000 of amino acids. The biggest known protein of the human organism is titine with 30,000 amino acids. The peptide bond is an amine linkage in which the nucleophile amine group of amino acid 1 attacks the carboxylic group of amino acid 2, and as a result they bind together. Water is released from the bond formation, which is why the reaction is called condensation reaction. The group taking part in the bond are a planar level and are characterized by mesomerism. This actually explains why hydrogen bonds are possible between the amino acid of a protein, which constitute a vital prerequisite for protein folding, as we shown in the figure. Mesomeric structure of the peptide bond. The end of polypeptide or protein which has a free amine group goes by the name N terminus respectively. The end with the free carboxylic group is called C terminus. In nomenclature, correct molecular names are produced when one reads from the N terminus to the C terminus, so that if the sequence is altered, a different molecule is represented. The virtually endless number of a possible combination is calculated according to x to the power n, where x is the number of different amino acids and n is the number of amino acids in a chain length. Protein structure principle or the overall structure of a protein can be very complex issue. In order to facilitate visualization, 4 levels are taken into consideration, which illustrate a gradual zoom out from primary to a quaternary structure. Now we are coming to discuss with you primary structure, looking deeply in the protein. The primary structure of a protein includes its amino acid sequence, namely the type and succession of amino acid in the fully peptide chain. Example are glycine, alanine, tryptophan, glutamine. Secondary structure, also called sectionally waved. Neighboring sections of the peptide can actually interact with each other. Their spatial array is described in the secondary structure, where there are many different shapes. The most important include that of an alpha helix structure, where the chain is right-hand coil or simply right-handed. A coil contains 3 to 6 amino acids. The inner part is formed by the main chain, whereas the side chain protrude externally in a spike-like manner. The helix structure is stabilized by intramolecular hydrogen bond between the NH group of the peptide bond and the CO group of the 4 subsequent peptide bonds. They are opposite each other in the coil. To the cause charge side function rather unfavorably, multiple helix can congregate in tangle branch or merge in so-called coil structures. Another type of the common secondary protein structure is the beta fleeted sheet, where the peptide bond is always planar and the chain follows a front and back zigzag pattern. The side chain protrude interchangeably from the sheet in downward or upward direction, which is the reason why they must be relatively short. Distinction between parallel and anti-parallel fleeted sheet is based on the direction of the N and C terminal end. The anti-parallel array, in which the N terminus is opposite a C terminus, is definitely more stable. In contradistinction to the helix structure, intermolecular hydrogen bonds are developed here. Peptide chains are always folded in a way that allow for a maximum number of hydrogen bonds to form. Now we are coming to discuss with you tertiary structure of the protein in the whole chain. The tertiary structure depicts the spatial structure of the complete peptide chain. We can distinguish various shapes, e.g. helix structure, fleeted sheet structure, or loops, or unclassified irregular section. They are formed as a result of interaction and bond which are mostly developed between the side chain. Now we are coming to discuss with you van der Waals forces. So what is van der Waals forces? Van der Waals forces are as a hydrophobic interaction, e.g. between non-polar side chain, hydrogen bond between specific or polar side chain, ionic bond, e.g. between the amine group of basic amino acid and the carboxylic group of acidic amino acid, particularly stable covalent disulfide bond between the two cysteine residue acids, through which the so-called loops are formed. Ester bond. During the denaturation of protein, their tertiary structure is always lost. The existing bonds are destroyed and new ones are formed, causing the initial protein structure to possibly lose its properties and function. Quaternary structure, a big functional unit. Big protein are composed not only of fully peptide chain but also by various subunits. Altogether they form a functional unit. Additionally, they can also form bonds with a carbohydrates, heterocyclic compound, and other molecules, a fact that underline the variety of their function. The interaction they have already been explained in the section for tertiary structure occur again here, and this time between the individual subunits. Thank you, dear ladies and gentlemen. If you like, please subscribe and share with others.